Okay, so I think the only thing we have left is hearing. The number one thing I saw in a lab practical that made me kind of sad was that I said, hey, I'm going to tag the vestibule. And it has nothing to do with hearing. Semicircular canals and vestibule have nothing to do with hearing. And so the number one wrong answer, which was repeated about 10% of the time, was that the vestibule is for hearing. And it has nothing to do with hearing. And that's what I said in my lab. It has nothing at all to do with hearing. It is basically movement such as just gravity and seeing which way's down and which way's up. And we have that big plate with the otoliths membrane and otoliths rolling back and forth on that. So we need to review semicircular canal and vestibule today and here also. So hearing. What's the purpose of this? In the first place, this part of the slide is just to talk about how sound waves are just movement, vibrations in the air, come in, and those vibrations in the air vibrate this bones, from the tympanic membrane, which is this big drum. It vibrates 3 bones. The one bone acts as a plunger and causes more vibrations of fluid in the cochlea, the only part that has anything to do with hearing, the cochlea. And those sounds are picked up by this little sheet of paper, basically, and we pick up action potentials. And so let's take a look at that. Again, there's high and low pressures, which just means waves in the air. And this is created with any kind of object moving. And so, like banging stuff, and anytime we hit things or make a guitar is simply a string vibrating, which puts off sound, right? And depending on how fast or slow and this thing vibrates, it gives us different sounds. So basically, we're just creating waves in the air, as we said before. So don't worry about refractions. I'm not going to put refractions and compressions, but it's just air moving back and forth. And so whenever that happens, it comes into your external acoustic medius. Now, when we look at the wave, we do see a couple things, and this is 2 things I want to mention. One is wavelength, and one is amplitude. You do have to understand these. Wavelength determines the high and low pitch. It's basically the frequency. How frequent do we have a wave? Does that make sense what I'm saying? So if these wavelengths are very close together, that's a high frequency, which you hear is like a lady singing at the opera. And then the farther they are apart is more like a bass. Amplitude has to do with volume. Amplitude is volume. So the more amplitude, the louder it's going to sound. The more amplitude, the louder. Now, we saw the tuning forks in lab. They have different frequencies, which have different sounds. And depending on how hard or softly it was tapped, that had to do with amplitude. And so it sounded louder. And again, frequency is the number of waves in a given point. The higher the frequency, the higher the pitch. The lower the frequency, the lower the pitch, more like a bass. And then, again, short wavelength. Am I saying this wrong? I think I said something wrong. This doesn't look right. Oh, no. Shorter wavelength means the closer they are together. The closer they are together. All right. And then, again, so frequencies go in different, they measure it by hertz. I have not ever put that on a test. I've never put pitch on a test. I've never put quality on a test. I've only put wavelength and amplitude are the only 2 that I know of here. And so, again, amplitude is how loud it is. Does that make sense? Just how loud. And it's normally measured by decibels. Now, here's something that I've asked sometimes. You may see it. But severe hearing loss can occur with prolonged exposure at 90 decibels. So in Indiana, we have something called the IOSHA, which is Indiana OSHA, which is Occupational Health and Safety Association. And so what they say is that in industry, you have to wear hearing protection, or at least it has to be supplied by the employer at 90 decibels. So anything 90 decibels and above causes hearing loss. Does that make sense? Sometimes I want to go get a decibel reading meter because there's, a lot of times I go to church and I think they're in the 130s, 140s sometimes. You leave and your ears shouldn't be ringing after church. It depends what church you go to, but some of them rock out, but others, it's might, get above 30 decibels maybe. But definitely rock concerts, anytime you're sitting near the speakers, you are prone to damaging your hearing. Okay, so let's get to the key points. We talked about the tympanic membrane. That's simply like a drum. It's just like this big dish of tissue inside your ear after the external acoustic meatus, and the sounds are coming in external acoustic meatus picked up by your ear. The bigger your ear, the more you can hear, right? And when those sounds hit it, it vibrates. And then there was 3 bones in order. The malleus is attached to the tympanic membrane. So in lab, this was a big thing that went wrong. I don't know what happened, but the malleus is attached to the tympanic membrane. And I think the thing that goes wrong is some people say, well, there's a hammer and an anvil and a stirrup, and the one that looks like a hammer is the malleus, and that's actually not true. The one that looks more like a hammer is actually the incus. So I tell people, ignore that. Don't listen to that at all. The one that's attached to the tympanic membrane is the malleus. The next one is the incus. And then the stapes looks like a stirrup or a staple. The stapes looks like a staple. And the stapes has an oval bottom, and it covers an oval window. And it acts like a plunger. Now, you would not believe how many people got malleus and incus and stapes mixed up on the lab practical. That was a big thing there. But that's the order. Malleus, then incus, and then stapes, and the malleus is touching the tympanic membrane. So, about 20 times larger than the oval window. So any kind of vibrations are going to be amplified 20 times. So just the smallest amount of sound that comes in, again, gets amplified 20-some times. There's a scala vestibuli, which is on the way in, and the scala tympani is on the way out. So the oval window is basically compression fluids. Those fluids travel into the cochlea, and remember, they go in a circle, and they come to a point where they have to kind of like turn around, because it's like the apex of the snail shell, if you want to call it that, of the cochlea. That's called the helicotrema. And so the helicotrema is basically that spot where it has to turn around, and then it goes to scala tympani on the way out. So vestibuli on the way in, tympani on the way out. The helicotrema is the port where it turns around. And so here we see the scala vestibuli on the way in, the helicotrema, and then on the way out again, scala tympani. But between these two, between them, we have the cochlear duct, as we said in lab. And the cochlear duct houses the hearing receptors, which we're going to see basically as the spiral organ of core teeth. So we have the tympanic membrane, the malleus, the incus, and the incus on the lab model looks more like a hammer than the malleus does, and that's why I have to be careful there. And then the stapes looks like this staple, right? It's oval shaped. There's an oval window under it. I don't think we mentioned oval or round window at all in the lab, did we? I don't think we did. But we do on the lab or lecture test. The oval window is the way in. The round window is right here. It's basically like a pressure relief valve, if you would. Because remember, the fluid's vibrations going in, it doesn't just stop. It has to have some way out. And so the round window is that basically the pressure relief. Does that make sense the way I'm saying this? You'll notice that these waves show that it deforms in a certain location. Bass and treble, I think this microphone speaker is, the battery's going out. But the bass and treble can hit in different areas here. And we'll talk about that in a little bit. But anytime that gets deformed along this pathway, it gives us an effect of bass or treble, or what, it depends on the frequency. But this is, a continuous thing that goes around in a circle until it hits the helicotremor. The one thing that we tagged in the lab practical is this on the way out, which is the eustachian or pharyngeotympanic tube. A lot of people label that the external acoustic meatus. Now that external acoustic meatus, again, is leaving over to the ear, so that's not the same thing. This is sort of any fluids in here can drain. Equalization of pressures, where if you go up or down a hill, the pressure inside has to equal the pressure outside, or else this seems like it pushes on the tympanic membrane and you can't hear. You notice that when you're traveling in a car and you go down the side of a mountain, things sort of get quiet, or if you're landing an airplane and things kind of get quiet, it's because there's more air down low and it's pushing in, and we have to equalize pressures, and you can usually do that by like holding your nose and blowing, opening your mouth, and so there's different ways of equalization there. Resonance. Resonance is a term that we use again in the chapter for respiratory. And what this means is like it's a certain vibration that changes the way sounds are. And that's what your sinuses help do. And so if you hold your nose, your voice sounds differently, right? And so nasal passageways and your sinuses are to help add resonance, and that's basically the way it vibrates. All right, so again, the cochlea. If you were to stretch this thing out, you could see it's actually quite long, but there's different frequencies that we hear in here. We hear closer to the base here, there's shorter or stiffer fibers, and then towards the apex are longer fibers. And so we're going to hear different frequencies in different places here, depending on where it hits along on this cochlear duct. And we'll get into that a little bit more. The inner hair cells, as we're going to take a look at here, we can see there's an inner hair cell here, and there's a outer hair cell here. And so the inner hair cells contain microvilli, we just saw right here. Oops. And basically, when those things are, I'm going to use the term create an action potential, we send that to the brain. And what we see about these cells is that basically because they have the tectoral membrane on top of it, they're picking up any kind of movement on the tectoral membrane. Now the outer hair cells, where is it? The role of the outer hair cells are a little bit different. They do fine-tune responsiveness of the inner hair cell, and they project inner hair cells, protect inner hair cells from loud noises by decreasing the motion. So what is thought is that the outer hair cells have a little bit less to do with hearing than the inner hair cell. Does that make sense what I'm saying? So the outer hair cell, it does help with hearing, but it also helps protect the inner hair cell because it's helping to hold and stabilize this tectoral membrane in place. But the inner hair cell is the more sensitive, the more one that has more to do with hearing than the outer hair cells. The outer hair cells and these cells on the outside are just called supporting cells, just to hold it in place. But they do help protect the inner hair cell, and the inner hair cell is the one believed to be more with hearing. All right, so don't worry about this auditory pathway. I'm not asking any questions here. There will not be anything here at all. Don't worry about that at all. The only thing I want you to always remember is that things go through the thalamus, because that's the mailman. All sensory information except for smell will go through there. So you can skip a big chunk of this. But this little picture on the side, again, the vibrations in the scala tympani and the scala vestibuli, when they vibrate here on the cochlear duct, and therefore vibrate on the endolymph and vibrate the tectoral membrane, as we talked about in lab. How do we hear different pitches? Again, this has to do with different frequencies. Frequencies are picked up in different places, and we'll go over that just more here in a little bit again. But localization of sound is this. If you close your eyes and you hear some sort of a noise off the side of your head, you can tell if it's right or left. Now, why is that? And that's because the noise is hitting one side before the other side. So the action potential on one side sends a signal to the brain before the opposite side because you're hearing it on one side. And therefore, you hear it on one side first, the sound comes around and then goes in. And so there might be a half second delay or a fourth of a second delay. And that's how we actually know where sounds are coming from. So if you have one ear that you cannot hear with, you definitely would have a hard time noticing where sounds are coming from. Does that make sense? So it says, depends on relative intensity and timing, relative timing of sound waves reaching both ears. If the timing is increased on one side, the brain interprets it as coming from that side. Equilibrium. The one thing that we want to talk about this is the vestibule, and this is the semicircular canal. When we talked about the vestibule, we're talking about more static equilibrium, basically more if you're sitting still and your body rotates in a certain position and these plates move back and forth with otoliths on it, we can tell which way's up and down. Semicircular canal, we said dynamic equilibrium or spinning. The other one that can be picked up on from the vestibule is what's called like acceleration from a car. You're not spinning, you're not rotating, but you're being pushed back. So it's going to mimic gravity in a different direction because you're being pushed in a certain way. Does that make sense? If you're in an airplane, like those fighter jet guys, they say they pull so many G's because you're being pushed to a certain direction. What's being acted upon there is the vestibule. Again, the semicircular canal is more big motions, like spinning and such. All right. So again, the cochlea, hearing, vestibule, static equilibrium, and semicircular canals, because there's three of them with the XYZ axis, have the dynamic movements, like spinning and such, and quick movements. Remember that we talked about this, and we didn't put it very deeply on the lab practical, if at all, but the receptors for hearing, the macula. Now, the confusing part about this is that we have a macula lutea, which we saw in the eye, and then this is the macula, more like a macula for hearing, but they're different name here. So anyway, the thing that's monitoring static equilibrium in the vestibule is the macula. And so key role in posture, again, which way's up, and also linear acceleration forces, like if you're in a car and they accelerate quickly, picked up by the vestibule. Not rotation. That's semicircular canal. Semicircular canal's organ of hearing, of creating the action potential is the crista ampullaris. The crista ampullaris. These are some questions I want you to understand for the test. The macula is in the vestibule, the crista ampullaris in the semicircular canal. Let's go back to the vestibule for a second. When we look at the macula, we look at the otolith membrane and covered with these little stones called otoliths. And basically it says that they are, the macula is horizontal with the vertical hairs, and as you change position, like you lean back and forth, they're going to lean. And so any forward, backward, side to side, anything that is going to change which way is up and down, this thing is going to move. And so let's take a look at that again. So here we see this hair cells, basically on the vestibular nerve. Now remember, when you looked at the ear, let's see if we have a picture of it. We have the cochlear nerve coming off the cochlea and the vestibular nerve coming off the vestibule. They will come together as the vestibular cochlear nerve. In lab, we had it a little bit separated. We had the vestibular on, tagged, or the cochlear tagged. We've no time tagged both at the exact same time. Here it is. This is another one that's a little bit different. We talked about like a car accelerating, but we also noticed that like on an elevator, you can tell if you're going up or down on an elevator by how it's pushing on you. And so if it's pushing on your feet or you feel like it's dropping from under your feet, we feel up-down accelerations the same as we feel front-back and side-side accelerations, all picked up by vestibule. So anytime we lean, so if we just turn, lean our body sideways, these things are going to basically move. The entire otolith membrane is going to move, and the otoliths. In lab, I talked about car accident victims, falls. Saw a elderly person had a bicycle accident the other day, and anytime they hit their head, they could do a couple different things. So you remember the superior sagittal sinus. Because of gravity, that thing pulls, and they could have a superior sagittal sinus tear. It happened to my grandpa. It happened to Jimmy Carter. He's been hospitalized for that recently. And they get a superior sagittal sinus tear, and therefore cerebral spinal fluid bleed, and that fluid builds up. But the other thing that's not huge traumas like that, but can happen inside the inner ear, are these little stones can get knocked loose. That's not something you would see. You'd look at the person, you'd say, are you okay? You do the cranial nerve exam, everything looks okay, blood pressure looks okay, everything's fine. And they go home, and then they start over a couple days, start noticing they feel dizziness. Especially when they're laying down and they start to turn, and the room will start to spin. And so what happens here is that one of these otoliths that got knocked loose will just basically tumble, and when it tumbles, it activates these stereocilia cells. And it'll basically rub right along these. And the brain gets the action potential that you're currently turning. And it's trying to figure out which way is up and down. And it gets kind of sent the wrong signal, and you start to feel like you're moving and spinning. And so that is one thing that can happen and can go wrong here. And again, picked up by the macula here in the vestibule. So the hair cells are going to release neurotransmitters, as we said, for action potentials. And any kind of acceleration, deceleration, or movement will be picked up from that. Ignore the rest of this. Well, yeah, ignore that. So anytime you bend those hair cells, you depolarize and send an action potential, and therefore it sends a signal to the brain that says, hey, we're moving. When you stop moving, it stops sending action potentials. So if you lean forward or back or side to side, the otolith membrane will move and therefore send an action potential. Ignore the rest of this. No, let's go here. So let's move on to semicircular canal. We're out of the vestibule. Semicircular canal. I need you to understand the physiology of this. We're going to ask a question about this. The crista ampullaris is now the place. I had used the analogy of stirring tea in a picture. So if you take a ladle and you're stirring tea because you put a bunch of sugar in there and you want to mix it up, once those fluids move, and they move in one direction, in a picture of a tea, it's spinning, let's say, clockwise. When you suddenly pull the ladle out, the tea still spins. And that's why people get dizzy, because basically when you rotate and twirl the body, the fluid starts to move exactly in one XYZ axis, depending on which way you're spinning, and it will continue to spin, and it's going to push on something called a cuplia. And the cuplia has these hair cells and stereocilia cells. And so this endolymph, as it's coming through and spinning and pushing, it will continue to push on that, and you will continue to get the sensation that you are spinning. The best way out of that, and here you can see an electron micrograph of that cuplia and how fluids come through and push on it. But if you were to go the opposite direction, you stop the spin. Does that make sense? And so when you see a figure skater spinning in circles, their next move, if they're going clockwise, their next move is always counterclockwise or vice versa, coming out of that spin. When the head rotates, the fluid spins, and the impulses are activated by the cupula. The endolymph will come to a rest, and therefore, you have now noticed that you've stopped spinning. But for a little bit, if you get it spinning a lot and suddenly quit, you will feel that spinning sensation for a while. Again, the endolymph kind of going over the top. Now, they drew the arrow backwards, and I'm not sure why they say that. It says, as rotation movement slows, then the lymph keeps moving in the same direction of rotation, which would be this. Endolymph flows, bends the cupula the opposite direction, would inhibit it. And that's what we said about stirring backwards. In other words, if you go clockwise and then go counterclockwise, it would do this. but they don't show that. They show a person stop, which is a little confusing. Does that make sense? So this doesn't really match the picture, is what I'm saying. All right. So when someone looks like they're spinning and comes to a stop, and they feel very dizzy, if you were to look in their eyes, sometimes their eye is still trying to follow the spin. And that term is called nystagmus. Don't worry about vestibular nystagmus, but nystagmus is eye movement during and immediately after rotation. And one of the things that we look for in this procedure called the Epley's maneuver is if a person has that dizziness, you turn them in a certain position, and they'll start to feel like they're getting dizzy, and you can watch their eyes kind of like trace the room as they think it's spinning. They will. The natural thing even on rides when you're on a carnival ride is not just to watch everything spin, but to like focus on something and then follow it, and then focus again and follow it. And you'll see them doing that even though they're not moving or doing anything. They're sitting still, but you'll watch the eye kind of move and do that motion because they're feeling sick and they're trying to watch things move so visually they don't just feel the spin. Visually they're experiencing that. So there's a mismatch between what the vision is actually picking up and what's being sent to the brain. And that's why a lot of people feel nauseous when this happens, because there's a mismatch of communication happening here. And a lot of people start to get really, really sick. And so as rotation begins, the eyes will drift in the direction opposite to rotation. For example, if you're rotating this way, you're not just going to follow it, you're going to try to look ahead of it and keep looking ahead of it to get things to kind of stabilize. That is the natural tendencies. As rotation ends, the eyes continue in the direction of spin, then jerk rapidly in the opposite direction. I'm not expecting you to know which direction they spin. Just know the nystagmus is that eye movement associated with that. We're not going to put what direction they go, or no, that's crazy. I'm not going to do that to you. Okay, so balance has three different inputs. Again, I don't think I'd even ask this, but it says vestibular receptors. In other words, what do you feel? What do you see? And and then this one, the soma receptors. In other words, proprioception. So where's your arms and legs in time and space? Anytime you mix those up, you get the car sickness or the motion sickness. So we get input from our inner ear, our vision, and I guess, again, what's going on in the body. When there's a mismatch of this, that's when people start feeling sick. So equilibrium problems usually are unpleasant and can cause nausea, as we said, dizziness, and loss of balance. There are some things that go wrong with the human body, and one of them we're going to talk about is motion sickness. And so some people have this, some people don't. Now, it's amazing how what you can be told can affect this. I've never felt motion sickness in my life. My little brother, however, always seems to get motion sickness. And I said, hey, you want to drive? He goes, I don't feel so good. And I go, well, we'll make you feel better. He goes, can I drive? And I go, sure. So I lay in the back seat reading the newspaper. I'm just laying there bouncing around reading the newspaper. And he said, oh my God, I don't see how you can do that. And I'm like, it doesn't bother me a bit. And I said, so what do you notice when you get car sick? And he started describing everything he noticed and how the things on the side of the window, he'll see everything going by and he'll feel the balances. And he started describing how if you're reading, he goes, those words be all jumping. And I'm like, pull over, I'm sick. I never got car sick before. But when he started describing everything, I start looking around and noticing it. And that's when that mismatch of communication came in. And I was immediately car sick. And I'm like, dude, you just ruined me. But at the time it was happening, I started paying attention to it. And it's amazing how you could mentally kind of make yourself feel sick if you start paying attention to these things. So what happens with car sick people is they tend to feel better with the window down. They tend to feel better in the front seat because they have a little bit better view of what's happening. Otherwise, you're putting them in a box. When you're in the back, you feel like you're in a box and everything's bouncing around. When you're in the front, you can see what's going on a little bit better and you feel a little bit better. Some people just, if you see them starting to bust out in the sweat, going pale, you don't want them messing up your carpet, you better pull over, right, or put the window down. They start breathing fast, the heart rate speeds up. Now, there are different medications out there. Meclizine is one that I've taken in the past, and I'm sure a lot of you guys have. It's just called, now it's over the counter. It used to be prescription. Now it's just Dramamine. So if you ever go on a boat ride or anything like that, there's a downside. Is it makes you sleepy. Some people say it makes you very sleepy, and I've seen first-time users take it, and they feel like they're just so sleepy they almost want to sleep on a boat. And so I'd say if you ever go on like an ocean cruise, try it ahead of time so that you can see how this affects you. The last thing you want to be is on a ship and find out you're allergic to meclizine or that meclizine makes you sick. But the other is once motion sickness starts, it's hard to stop. Does that make sense what I'm saying? So if you're like, hey, we're going to go on a cruise, take the meclizine the moment you get on the boat, even before the boat takes off. If you're prone to motion sickness, take it the whole time that you're on that boat. If you wait till you're sick, it does not help much. Not nearly as much. You got to kind of take it early. Anybody ever go on a cruise? The last thing you want to be is in like day 3 and puking, and you got like 4 more days to go. That is a horrible, horrible sensation. The one thing that I've noticed about being on a boat, and some that boated before might agree with this, is that when you get off the boat, if you spent a lot of time on a boat, like I've spent weeks on a boat, for days and days afterwards, you could be sitting at a table in a normal place and you're feeling waves. And so that's an weird thing that kind of happens, but it's just a input, kind of a confusing signal sent to your brain. And I've tried to look into that and figured why do you continue to feel waves for long periods of time? And nothing has been able to really explain to me so far. If you find a good article on that, I'm curious. Anybody notice that when you get, especially if you're in the ocean and waves? So that's another one. So let's go back to hearing for a little bit. We talked about different types of inability to hear properly. One was conduction deafness, and this could be caused by just ossicles, getting old, the earwax buildup, all these different things. But blocking the sounds of conduction because just the sound waves can't get in. And we did the Weber and Rhine test to kind of determine this. The sensory neural is the true nerves being damaged, the true nerve damage, and the true issue happening with the nerve cannot pick up that action potential. And this is one that would require more of a hearing aid, whereas a conduction deafness, it could be the eardrums damaged. It could be because the eustachian tube's not functioning properly, and they put a tube in your ear, and where they put a tube is in the eardrum, by the way. I don't know why we put eardrum instead of tympanic membrane, but it's there. Ear infections might require an antibiotic, removal of earwax. There's different things can be done there. Cochlear implants and hearing aids are two things that, help people. We have a friend that their baby was born deaf, and the mother came to me and talked about cochlear implants, and I said, look, I'm the wrong dude to be talking about this stuff. I don't know anything about it. But she Googled what a cochlear implant sounds like. And I just assumed it would sound like what you guys are hearing right now, but it's not at all. And if you go online and check that out, it's kind of a scarier sound compared to what you'd imagine. It's like somebody kind of growling and talking at the same time. It's a very rough sound, but they can hear it and they eventually decipher what those sounds mean. And so here's an example of a cochlear implant. So tinnitus. Anytime you hear a ringing in your ear, that term is tinnitus. Sometimes it could be loud, sometimes not, and that's going to lead to another conversation, which is called Meniere's disease here in just a second. But look at this one, side effect of aspirin. Also a side effect of blood pressure being too high. Sometimes they'll hear it ringing in their ears. And here's Meniere's. So the first time I ever heard about this was from a patient who said, hey, I have Meniere's syndrome. And honestly, I didn't know anything about it at all. And so I just said, I said, look, I'll be honest. I always say, state the truth, be honest, state the obvious, And I said, look, I've studied a lot of stuff in school, but I forgot about this one because I don't know anybody's had it. Can you remind me? He said, well, I feel dizzy a lot, and I have this really loud ringing in the ear, And I said, well, how bad is it? And he said, it used to be so bad that if you would put your car or your ear on the hood of a car and somebody honked the horn, it can get louder than that. And when this guy had acute flare-ups of his Meniere's, he would have to basically write notes back and forth because he couldn't really hear very well, and he just didn't want to communicate. So sometimes it gets bad. There's some people had it to the point where they just can't really work. We had one of our coworkers then got it, and I just talked to them a while about it, and they said it's genetic. It can be genetic, and their uncle had it, and I guess another grandparent had it. But what happens is it can cause dizziness, and she got to the point where she had to hold on to the wall to basically walk. She just got that dizzy to where she'd walk and just hang onto a wall, and basically it ruined her career. And I'm not sure how she's doing, but the nauseousness, vertigo, the tinnitus, all kind of mixed together. And again, it could be genetic. So there are medications out there to kind of help with that. Some people lose the ability to smell, which we should not be talking about at this point because we have already covered it. But the term anosmia, I want you to remember as something that we mentioned that might be testable. This can happen with rapid adaptation or smells that are too strong or age. Sometimes people lose the ability to taste and smell. Remember that 80% of taste is from the smell. And that elderly people lose the ability to taste foods, they'll quit eating. You'll start seeing them lose weight, and they're not trying to do it on purpose, and you start thinking, oh my gosh, there's something wrong. One of the major things is they just quit eating because they just lose their appetite, basically, is the best way of saying that. All right. We've already talked about vision. Let's just review a couple quick things that might be on the test. I'm never asking any developmental things. But remember, on vision, you need to know the parts of the eye. You need to know that the cones are for color and crispness. And that term crisp vision is acuity. Some people mixed up what acuity meant on the test. But acuity is how sharp, how crisp. We typically see these in color vision. It's in the very back of the eye, the macula lutea, and the center of that is the fovea centralis. On the periphery, we still have a few cones, but not as many. We have more rods on the periphery. They're more grayscale. They're more fuzzy. They're definitely in low light conditions. You can see better with the rods than you can the cones. The rods is a combination of the opsins and basically have to build up something called rhodopsin. And rhodopsin takes about 20-30 minutes to build up, and that's why dark, to be able to see in the dark takes 20-30 minutes to build up that rhodopsin. If you walk out in the bright sunlight, it bleaches out. It physically hurts the eye. Like you could feel pain from bright sunlight because you're bleaching the rhodopsin. The cones take 3, 4, 5, up to somewhere in there, amount of minutes to accommodate to the light. I may ask a question about nearsighted versus farsightedness. Make sure you remember those terms. One eye is too long, one eyeball is too short. What type of lens can correct those? We'll probably ask a question about color blindness. And I have a question for you guys. So we're done with this chapter, and I want to ask questions about how you prefer to do things on our next meeting. We have